So this summer I had the pleasure of working in the Van Wyck lab under Eamon Bushnach, my mentor, and I worked on protein degradation in chloroplasts. So why were we interested in this subject? Well, we know proteins are integral to biological function in numerous ways. Just a couple could be cell-to-cell signaling or transport in the cell or structural support. The possibilities are numerous. And really what's important is understanding the timing of these processes. So when is a protein synthesized, when is it degraded? And my project focused on degradation. And why did we pick the chloroplast? Well, we know it's a prominent organelle in the plant cell system. It's the conductor of photosynthesis in the cell, so it's going to be producing energy. And it's vital to the actual cell's growth and development. So we kind of wanted to know a little bit more about how this pathway actually functions in that organelle. So, the end rule. In several degradation pathways in both prokaryotes and eukaryotes, there is what's called the end rule, and pathways function according to this. It basically states that certain amino acids, when exposed at the N terminus of a protein, act as signals for degradation of that protein. And in bacteria, these 4 residues are lysine, phenylalanine, tryptophan, and tyrosine. And stabilizing residues are alanine, serine, threonine, and valine. So basically that means that if any of these 4 are exposed at the N terminus of the protein, that kind of signals to whatever machinery, hey, this protein is ready to be degraded, come pick it up, get rid of it. And one important system that follows the end rule, because there are multiple, is the CLIP system in bacteria. And this system consists of an adapter protein, CLIP-S, which is responsible for recognizing proteins according to their N-terminal substrates, picking them up, and transferring them over to the CLIP-AP protease core. Now the CLIP-As are chaperone proteins that bookend the CLIP-P in the middle. The CLIP-S makes contact with one of the CLIP-A chaperones via its tail, deposits that substrate protein in the pore. That substrate protein is then degraded while the CLIP-S is recycled for future use. So, what does this have to do with chloroplasts? Well, we know via the endosymbiosis theory that chloroplasts were derived from cyanobacteria. So that kind of gives us our first jumping off point to how bacteria and chloroplasts might be connected. Next, we know that a CLIP system actually does exist in chloroplasts. More importantly, in 2013, my lab did discover the CLIP-S1, they named it. It's a possible functional homologue of the CLIP-S adapter protein I talked about on the last slide, and it's really important to my project this summer. So that kind of gave us another slice of the evidence pie, if you will. Also in 2015, my lab did another study of stromal proteins in the chloroplast. This was just analyzing what N-terminal residues are present most often. And what they saw overwhelmingly was a mirror of the bacterial stabilizing residues. Those were alanine, serine, threonine, and valine. So you can see the bigger slices of these pies, and it was normalized, which you can see in the C figure for just how much of them were present all the time. And the same was true for the rare residues, which in this case were lysine, phenylalanine, tryptophan, and tyrosine, which were the same as the destabilizing residues in bacteria. Lastly, in 2019, my lab also did a study, an affinity assay with recombinant CLIP-S1 and possible N-degrons. And what we saw from this, you can see here in this, it's kind of hard to see, but in this lane here with CLIP-S1, we've got a very dark band for tryptophan and for phenylalanine, which just showed specificity of binding for both of those amino acids and that recombinant CLIP-S1. So that kind of gave us 2 possible candidates for our assay this summer as test N-degrons. So what were our hypotheses? We hypothesized that an NN rule does in fact function in chloroplasts, and not only that, but that the CLIP-S1 is essential to this in substrate recognition and delivery to the core. So our objective was to develop a system that would allow us to test this idea. So we came up with a fluorescence assay, and this is the basic setup for that. So we've got a protein here, you can see tagged with GFP, which just kind of marks where it is in the cell for us. We need this to be happening in the chloroplast. So this will signal where this protein actually is in the cell. And since we want our assay to be functioning in the chloroplast, we've tagged it also with a chloroplast transit peptide here. And that chloroplast transit peptide will direct this protein to the chloroplast after it's transcribed and translated in the cytosol of the plant cell. And after being imported via machinery on the chloroplast membrane, this tag is cleaved. And it's cleaved by an endogenous stromal processing peptidase. Endogenous just meaning that this protein is native to the plant cell. We did not need to introduce it with a plasmid or a transformation. It's already there. And it recognizes that chloroplast transit peptide, and it swoops in, and it gets rid of it. Says goodbye, we don't need you anymore. So you're probably wondering how we're going to actually test our integrons, or our possible integrons. How are we going to expose those? How are we going to do it at the right time? Well, we decided to use TEV2 protease under inducible expression, which just means it's under our control for when it's transcribed. And this protease has a very specific cleavage site. So TEV2 is able to come in, recognize a specific cleavage site, and cleave our protein right there, thus revealing a chosen N-terminal residue that we have marked right after that cleavage site. For our GFP protein, it's the purportedly stable alanine, and for our other protein, which is an RFP, it's the supposedly destabilizing tryptophan. And based on this, we can just kind of compare the fluorescence between our GFP and our RFP proteins and see if this system is actually functioning as we expect it to or hope it to. That RFP should be recognized by the CLIP system and degraded accordingly. So we should see a sharp decrease in RFP fluorescence over time. So here is our starting point, our plasmids. I wasn't here for this part, but I'm going to speak a little bit about it because it's important. You can see the TEV2 plasmid over here, and like I mentioned, it's under inducible control. That's very important because we need to make sure we know when our assay is actually starting and when this cleavage is happening and when the fluorescence is decreasing. So how this inducible control works is we've got a 35S promoter that's in charge of this LHG4 transcription factor. That transcription factor, that protein is kind of out of commission because it's bound to an HSP90 protein. And how we liberate that protein is by administering dexamethasone to our system. That dex liberates the LHG4 transcription factor. That transcription factor then binds to this LAC operon, which is in charge of transcribing our TEV2 protease. So until we had put dex in our system, there is no TEV2 protease being transcribed. So that kind of gives us control over when TEV2 protease is actually being made and when it's performing its function. In the middle, you can see our GFP and RFP construct. This is our tandem construct where we have just one constitutive 35S promoter, meaning both the proteins are going to be produced constantly in the cell. There's no control over when that's happening. And they're both produced in tandem, so in the same direction at the same time, and are cleaved apart later by TEV2 protease. You can see this one's got the alanine, RFP's got the tryptophan, and here in our last one, our independent or our single construct, it's very similar to this one, except we have two 35S promoters, just going in opposite directions, each transcribing their own fluorescent protein. So we took our plasmids and we transformed them into three different backgrounds, three different plant backgrounds. We first had to transform agrobacterium with the plasmids, and then we did the floral dipping method with our three different backgrounded plants with those resuspended agrobacterium. And the three different backgrounds are our wild type, which contains all the CLIP machinery necessary for degradation. So it's got the CLIP S1 adapter protein, and it's also got a CLIP F protein. And this CLIP F protein is just a posited co-adapter of the CLIP S. We're not quite sure what its function is yet, but we didn't knock that out in our wild type version, so it's still there. We've got a CLIP S1 mutant that does not contain any CLIP S1, but it does have the CLIP F protein. We've got our double mutant also, which has no CLIP S and no CLIP F. So those were our three constructs that we had. We transformed into those backgrounds. We waited for our plants to grow seeds, and then we harvested those seeds. We then took our seeds and planted them on MS and hygromycin media to do our first selection. So our first selection, we just kind of looked at how healthy is one plant, little seedling compared to another, what's the height, versus healthiness or deadness looking. They look pretty dead, you can tell. So then we took our little seedlings, we put them on Cornell mixed soil in the center, waited for them to get a bit bigger, and then we took a leaf sampling and did a DNA extraction and a PCR amplification to actually select our transgenic plants. And here at the top you can see TEV2, we've got it in all three backgrounds, wild type, CLIP S1 mutant, and CLIP S, CLIP F mutant as well. And we're really just checking to make sure, is that band there? That's all we're looking for. Yes, it is. We know the plant's transgenic. Perfect. You can see our GFP and our RFP proteins on the bottom. It's kind of hard to see. They're a little fainter, but they're also there as well. So we take our transgenic plants, we know for sure, and we move on to our next step. And the next step is checking transcripts and dex inducibility. So we check DEX inducibility for TEV2, right? We want to make sure this protein's not being transcribed, it's not present until we tell it to be present. And we basically did this by we did a sampling of our leads before administering DEX. So in this line you can see there are no bands, there is no TEV2, which is what we would like to see because we don't want it to be there when DEX is not administered. We then sprayed our plants with a DEX solution and took samples 2 hours, 4 hours, and 24 hours after doing so. And you can see a smattering of different bands of different intensities. And reasons for that could just be that some leaves were maybe under others and didn't get sprayed fully, and that could be a reason why there's no band, or that the sampling was just too small to really get an accurate RNA sample. But really quantifying this and figuring out when's the best time to take a leaf sample is going to have to be done with qPCR, which we attempted, but we didn't really get a great readout from that, so we're going to have to do it again. Also important is checking transcripts for our GFP and RFP proteins. And this is just to ensure that any loss of fluorescence down the line is not due to a discrepancy at the beginning with difference in fluorescence, but only due to that N-terminal residue being present. And then last but not least, we test for chloroplast localization, just to make sure our proteins actually made it where we wanted them to make it. So we do this 2 ways. We do confocal microscopy. So here's our microscope slides you can see, and just a quick pic of our fluorescent protein. So we've got our autofluorescence of chlorophyll in blue, our RFP and GFP in their respective colors, and the superimposition in white. This just confirms to us, yes, they are in the chloroplasts, and we can move on with our experiments. We also confirm localization with immunoblotting or Western blots. This is especially important for TEV2 because it doesn't have a fluorescent protein attached to it, but we can see a mass shift of about 5.5 kilodaltons when that chloroplast transit peptide is cleaved. So, last but not least is our actual assay. This is just a theoretical model for what I would imagine it being. We didn't get to this point, but here we've got our GFP tag with our stable alanine and our RFP with our destabilizing tryptophan. And here we have an increase of these proteins as they're made, but as soon as TEV2 gets in there and it cleaves those proteins, we should see a sharp decrease in RFP fluorescence over time, whereas GFP will kind of plateau. So just a quick summary of what we've done and where we need to go. We have transformed our plants with TEV2 and GFP and RFP. We've genotyped those plants, we've checked transcript levels for TEV2, and we've also confirmed chloroplast localization for our GFP and our RFP plants. However, what we still have to do is test TEV2 chloroplast localization, check our transcripts for GFP and RFP, cross our plant lines, and perform the actual assay. And hopefully with that knowledge, if our assay goes according to plan, we'll learn a lot more about plant growth and development via chloroplast. These are all the people I'd like to thank. My mentor, Eamon Bushnak, who is on a much well-deserved trip home right now. And our PI, Class Van Vick, and then all the members of the lab, Julia Friso, Elena Michelle, and Michael Miller, who all helped me out this summer. Elena's over there. She's present. And Delaney Sickler, who's been in charge of us for these months and has done that wonderful job. George Jander, who's given us so many opportunities with this program. Meredith Williams, who's also helped out, I see you in the back. And then all of my fellow interns who've made this experience awesome. So, and all the funding sources too. So that's it. If you have any questions. Yeah, that me.
So, in what situations would the chloroplasts actually want to degrade proteins?
In what circumstances would it want to degrade a protein? Well, if you think about certain protein pathways, a lot of it can depend on upregulation or downregulation of the protein, right? So in any circumstance, you could have a need for a protein at a certain time and no need for it at another time, right? If part of the growth and development is due to a protein being present at a certain time, if you've moved past that stage of development, do you really need that protein anymore? I'm not sure. I'd say that's the circumstance where the plant or the chloroplast organelle might say hey, we don't need this protein anymore. It's not vital to this process at this moment in time. It's kind of just like feedback loops, right? There's a time when you'll need a protein and when you'll not. So that's why I would want to degrade them.